Thank you very much to the organizer for this invitation. It's a great pleasure for me to be here, because of you first, and because I've never been to Kobe before, and also because everything I will talk about today initially originate from Japan. So as Eric pointed, I will try to place this model description within something a bit larger, but mostly at the end of my presentation. I will not try to justify the need for what we're talking about here, and this presentation will be very technical actually. But I would like to restate something about model exchange. First of all, so models are now beginning to be at the core of integrative systems, biology systems, neurobiology, physiology, whatever. The model used to be just one tool among others. There is now a movement where the model is really the core of our activity, the big integrator. But I would like to emphasize the fact that as for any kind of output from scientific research, models must be available to the scientific community. And that's something that traditionally has not been the case in computation, neuroscience, and neurobiology. Basically, when you had a paper, you read the result, and you had to take the claim from the author at face value. And that's just not acceptable. A model that is not distributed cannot be verified, cannot be falsified, therefore it's not science. A paper without the model is equivalent to an experimental paper where you can't have the reagent and verify the result. So that said, and that's independent from standardization or anything. Right? Now, going to standardization, so computational models now must be exchanged so they are to be storable and readable. They must be related to relevant experimental data set if they have to be at the core. And because of that, we need expressive relationships, robust links, and a rich set of metadata that's been emphasized by Sten Gridner this morning. Those computational must be amenable to many different analyses, not just simulation, and we need standard API for that. And finally, those computational models must allow modification, split, merge. Just a few minutes ago, it was mentioned that we could change a single compartment description to a multi-compartment description. So for that, the model has to be encoded according to a suitable design. It's not random. So for all that, standardization is unavoidable. I mean, the debate is over. In systems biology, we've developed standard for a while now. And we have this matrix of standardization. As for every part of scientific research now, not just computational biology, you need 3 layers. You need a layer that describes what you need to exchange, the minimal requirement, and I believe there will be a presentation of the result by the Carmen effort that is dealing with that. We need standards to encode what you want to exchange. And finally, again, we need ontologies to put some sense on that. And until recently, we had 3 different types we wanted to encode in system biology: the model description, the simulation description, and the result of those simulations. And here you close the loop to experimental investigation, of course. And I will briefly describe 2 of those efforts because I think they're relevant for us here. The first one is SED-ML. It's been mentioned before. So what is SED-ML? The System Biology Markup Language. It's a way to exchange model and reuse and hopefully interface different models of quantitative models in system biology. Initially, mostly well-steered chemical kinetics. But this is evolving. So this is not a procedural language. It's very important to understand that. It describes the structure of model, not what to do with it. It's not a programming language. It is not a format for a specific software configuration. It's been mentioned before, but I want to re-emphasize the fact, putting some timescale here. Only 3 of the 7 SED-ML funding software are still in operation. And actually one of them, ESL, as far as I know, is only used by 2 groups, including myself. So it's really important that designing those languages, you need to disregard completely existing software, because in 10, 15 years, they won't be here anymore. I mean, even the programming language will be different. Okay, I will zap this. So this is the global structure of an SED-ML file, a series of lists of components, but mostly you have two types of components. You have variables, so compartments, species that are entity pool nodes, entity pools basically, parameters, initial assignment, and relationships that link those variables together. Okay? And as you can see, there is no semantics in the name. This is a very, very simple SED-ML file that describes one compartment called cell, two species, A and B, a global parameter, you could have local parameters as well, and then a reaction here that converts A into B. And importantly, it's described using a mathematical expression, and this mathematical expression uses an existing standard developed by the World Wide Web Consortium called MathML. And this is one important information. We use existing standards when they exist rather than reinventing yours. And each of the symbols here are equivalent to a quantity. So that was a very, very simple example. If you take this line here, a realistic version will be that. It says that this species with an identifier A is alpha tubulin, it belongs to the compartment cell, it has a certain amount, certain properties here, and again, standardized information, metadata for human consumption encoded in XHTML, another standard from the World Wide Web, and for software consumption in the resource description framework, another standard from the W3C. Okay. So SED-ML is not limited to biochemistry. That's something we see again and again and again. Everyone mentioning SED-ML this morning mentioned it as a language to encode biochemistry. This is just a myth, right? We have rate rules that can describe temporal evolution of any quantitative parameters, discrete events that can, well, describe event. And the species is actually any set of identical elements reacting the same within the context of a model. So it can be a molecule, population, a cell, organ, organism. SED-ML, what SED-ML is, it's about process description. It can describe any variable that evolves according to processes, right? So in the course of this presentation, I will talk about models. You can find all those models in biomodels database mentioned by UP before, right? And this kind of database are only possible because of the standardization. I mean, they're pretty different from ModelDB. Of course, you can encode signaling pathways, as described. So this is a model integrating the stimulation by dopamine through a cyclic AMP and stimulation from glutamate receptors through calcium. And you can follow phosphorylation of proteins here, different phosphorylation state of DART32, and put perturbation here, CMP and calcium spikes. That's the paper, okay? And that's a reproduction by the curator of biomodels database. So, because in a different software, sorry, very important to say. That was done in eCell, that was done in SED-ML solver. So, as I already presented how it worked, you have different species. Here you have two reactants, DART32 and CDK5, and they assemble to form a complex, right, according to this reaction with a velocity described by this mathematical expression. You can also encode conductant-based description, so that's the Hodgkin-Huxley model, right? This is the paper, this is a reproduction in SED-ML solver, and that's done using two different constructs, the rate rules that describe the evolution of the variable voltage according to the variable, and an assignment rule. Here, for instance, the sodium current in function of the conductance of the channel, etc. You can also encode single point models. Those are a neuron described in the famous paper of Eugene Izikevich. And those are the simulation of the model using a software called Copazi. And this is done using rate rules again. Here the voltage in function of the different variables and discrete event. So here I have a trigger. When the voltage is above the thresholding voltage, then I assign those values to the variable u and d. Okay? So we can do a lot of things with SED-ML that are not chemistry. Okay, so SED-ML is developed according to different levels that coexist, and the only message of this slide is that we progressively simplified, generalized, and externalized information in SED-ML. And that has been done because over the 10 years of SED-ML development, there was a shift in the development of SED-ML. We started as a community of biologists developing a standard for our activity. So we were developing software, we basically designed something, and we learned how to design a language as we went along. This is not the case anymore. Now, people developing SED-ML and SED-ML supporting software are computer scientists, software engineers. They are not biologists. They take input from biologists, but it's more professional, if you want. SED-ML level 3, the last version, will be a modular language. So there is a core package that everyone must support. And then you have modules that different software sharing the same representation can share. And I think that's an interesting model to follow. Maybe not the only model to follow, but this idea of a modular language may ease the path to implementation, not forcing everyone to implement a super huge language, because that doesn't happen in practice. People just don't implement what they don't want. So after SED-ML, I said SED-ML was storing the model description, and it's been said by Eric already that this is different from simulation. It was emphasized when talking about 9ML that simulation description was dealing with software and was something different. And to do that, we have another language that started in Okinawa 2 years ago called Simulation Experiment Description Markup Language. When we designed the minimal information for the annotation of, required in the annotation of models in 2005, there was a funny rule, the rule 6, that says basically when you run a model, you have to be able to reproduce the result. But it didn't say much about the mean to do that, and SED-ML or SEL-ML do not offer any particular information to do that. And if you look again in models in biomodels database, we provide a screenshot of the validation of each model. Basically, all the model we succeed to run and reproduce the result. And if you look at those representations, they're actually hard to reproduce if you don't know how they've been done. This one is very easy, so that's a neurotransmitter receptor. You just plot the x-axis in a logarithmic fashion, quite easy. Here you have a double oscillatory system. In order to reproduce the phase plane image, you need to know which variable to plot against which variable. It's a bit harder. Here, it's a model of map kinase cascade. And now you need to know that you have to do a parameter scan of a given variable. So here we run 100 different models with different initial values. If you don't know the sampling and the boundary condition, you can't do that. And finally, here you have a two-dimensional parameter scan followed by post-processing of the numerical result. So there's none of this information is in SED-ML, right? So in order to plug this hole, we developed first another MI, the minimal information about a simulation experiment, that lists everything you need to exchange, all the information you need to add to your article so that someone is able to reproduce the simulation, and the corresponding language. So the corresponding language is structured as followed. You have the list of the models you will use in your simulation experiment, the list of the simulation approach you will use, the list of simulation tasks that will link the simulation approaches to the model, then how you will generate your data and how you will output them. So quickly, how to describe a model. You can load a model and then you can change this model in order to launch your simulation, change single values or compute some change based on other variables that can come from other models. So that's a small example. I load a model. That is the model of Izikevich giving regular spiking. This model is in SED-ML, but it can be in any XML language. SED-ML is independent of the language you use to encode your model. The model is coming from Biomodels database, so that's called a Miriam-Muren. It's a standardized way to encode data set in systems biology. And using that, I can retrieve the model automatically using web services, for instance. And then I define a second model. The source is the first model, and I modify it using those two XPaths that tells me to change the value of C and D to new values, right? And that will give me a chattering neuron. Then I choose my simulation approach. Here, this will be a uniform time course. I basically give the simulator the information about starting time, output time, the number of points I want. And again, an ontology tells me which algorithm to use. And because it's an ontology, if I cannot access this algorithm because my software doesn't support it, or because that was done in a proprietary software, using the ontology, I can find the closest algorithm available to me that I can use. Finally, I link the simulation to the model. Oops. And I will describe the generation of the data, which variable from which task to use, and how to post-process it to get the numbers I want. So here I create 3 data generators. One is a time, the other is a voltage, and the third one is a current. Except the variable time that is standardized by SED-ML, the others are obtained using those strings that are called XPath. It's another World Wide Web Consortium standard that allow me to access one particular value in a particular XML file. Because I'm within SED-ML, that actually tells me the values of the variable described by this model piece. And finally, I can produce the output, either reports with plenty of variables or so-called plots, very badly named, that are not really graphical things, but that tell us which variable to report in function of which variable. So we have 2D and 3D reports at the moment with various information such as the log scale and so on and so forth. And here I go. So this is my regular spiking neuron. I loaded the SED-ML file, and I tell the simulator, run the model, and at 10 seconds, change the current, and it triggers the regular spiking. Okay. Now, quickly, that was my initial mosaic of standard. That was only for systems biology, and the question is, how complete is that? Because that was complete in 2007, the first time I made this slide, but this is not the case anymore. First of all, we need to disentangle the model lifecycle more finely. And I think that was hinted in the previous presentation during the question. Describing the model is fine, but you need more. You need to describe how to generate this model, and you need to describe how to parameterize it. Where did you get the number you put in there? How, from which data set, and which procedure you use to get from the data set from the parameter value. The model generation is needed because now in functional genomics, in toxicogenomics, but in neuroscience obviously as well, you need to generate the model description from another source. I'm not talking about the initial values here. I'm talking about the structure of the model. You have people running Bayesian network, for instance, who generate chemical kinetics models from cell culture. So those we talked about already. You need to disentangle the level of discourse, and that was described a bit in 9ML as well. You have the model semantics, which is the structure of the model, the list of variables, and the relationship between the variables. That will be the abstract layer of 9ML. Then you have the initial conditions, the numbers, because you may want to use the same model with different numbers coming from different experiments, for instance. You have the biological semantics, and that will be the user layer for 9ML. And finally, the graphical representation. So we have a few descriptions in system biology for that. The initial conditions at the moment are merged with the model semantic in SED-ML. That was known from the very beginning to be very, very bad, but we wanted something working quickly. And finally, there is a problem of parallel and redundant effort. So that's a very important message I would like to convey here. This is why I will overspill my time. There are efforts in biochemistry, system biology if you want to call it that way. You have efforts in physiology, neurobiology, in drug discovery, in ecology. Those efforts are starting all the time. So at the moment, of course, you all know the efforts from the INCF. You may have heard, some of you, about the virtual physiological human, which is a European project aiming to develop yet another set of standards. And this is a new effort just starting, spawned by the pharma industry and the academia in Europe, that aim to do exactly the same from drug discovery models, pharmacokinetic, pharmacodynamics models, very different type of model. So the question is, what if the World Wide Web was built like this? You know, scientific, developing their own representation language for science, people doing literacy, their own business, art and design, leisure. Everyone has his own standard describing the web pages with their own. And it started actually like that, if you know the history of the XML. But this is not what has been done at the end, of course. The correct way to do it is to have one language for the display of information. That's called HTML if you want. Graphics, SVG, semantics, ODF, presentation, the cascading style sheet, personal information, vCard, and so on and so forth. One language for one type of information, one structure. So what we want is actually one language for biochemistry for process description, one language for morphology. At the moment you have two languages in your ML, morphology and state transition, but that's really two different things, MorphML and channel ML. One for mechanics, there's nothing in there. One for tissue network, and here I would like to propose a challenging idea that maybe 9ML is not specific to neuroscience, because developmental biologists, plant biologists use the same kind of models. They have networks of cells, and really what we want is a language to encode tissue networks more than neuronal networks. And finally, a language for global representation. There is an effort in physiology called SED-ML for that, but so maybe it could be applied for a global voltage recording or EEG. That's to answer Sean and to stem discussion for the for the panel. Maybe we don't want one language that encodes everything, but plenty of language spanning different scale. And what we really need is a variable transformation language, an adapter language that will tell us, use variables coming from this model, apply those transformations and spit out other variables. Maybe SED-ML can be used for that at runtime, during the simulation time. But anyway, so there are efforts in system biology to interoperate between the different standards. SBML to BioPax can be converted. SBML and BioPax can be converted to SBGN, the graphical language. SED-ML can use SBML, CellML, VCML description in the same way. It's independent of the language. And we started, NeuroML and SBML started to discuss in order to interoperate. So there are something going on. I will zap that. The idea here was to justify the existence of an overarching structure, and this is what I will describe now to finish. So after 10 years, we have all those efforts. They are all interoperable. The same people are involved in the development of those various efforts, and time has come to come together and stop working independently. So this is why we created Combine, the Computational Modeling in Biology Network. And this Combine aimed to become the World Wide Web Consortium of Modeling. The first step was a series of meetings that started in Okinawa in January 2008 when SED-ML was born. And progressively we merged all our meetings to come to the first Combine meeting in October this year with SBML, SBGN, Biopack, SED-ML, but people from NeuroML will be there as well. And that will be, from now on, the unique meeting every year. And there will be another type of meeting, a more technical meeting in spring every year called Harmony that will bring all the developers together to do actual work rather than talking. So the first meeting, as I said, will be in October in Edinburgh. We have so far 75 people who will attend, 14 sessions covering the various languages, but also issues like format conversion, metadata, the resources, semantic and metadata resources, how to encode metadata and use it. And it will be followed by the 10th anniversary of SED-ML. And I would like to finish by acknowledging a few people, but such an acknowledgement slide is totally impossible to design because we have literally hundreds of active contributors and 1,000 people who kind of contributed. But the person to acknowledge is Professor Hiroaki Kitano, who actually launched the effort toward SBML, launched the effort toward SBGN, and he is pushing all those efforts. The SBML editors, the SED-ML editors, so it's very important that I mention that all those efforts are community-based. The editors are elected by the community, and all the decisions are taken using community votes. And finally, I would like to thank the whole community of computational system biology and my group, of course, who is doing the actual work. Thank you.
Hi. Hi. Thanks very much for the talk. I'm here. Hi. Hi. So you mentioned SBO, the Systems Biology Ontology.
Yeah.
I just wondered if you could say a bit of, is that useful in the biomodels database for taking models and putting them together? And do you think it would be an oversight if some provision for an ontology were left out of a modeling description language?
Sorry, last part was?
Do you think it would be an oversight if some component of an ontology was left out of a modeling description language?
So the answer to the first question is at the moment, no. Biomodels database doesn't rely on SBO term. You can use them for search, but we don't use them. However, SBO is used to convert SBML to biopacks and convert SBML to SBGN. And people using, developing tools like Semantic SBML that allow you to merge models are using SBO. Now, the same people are developing a new search engine for biomodels database that is based on clustering the models. And this is using SBO, but it's not in the public version of biomodels database yet. We just submitted the paper and we need to brush a bit the software. The second part is, I think with the proper API, we could left out some information from the model if they were stored in ontology. So at the moment, this is not the case with SBO. SBO tells you the kind of parameters it is, kind of species. There is mathematics in SBO terms. So theoretically, if you have the proper software, you can read a kinetic law, the SBO term that correspond, and load the math from the SBO term rather than to explicit the model. But that's not really used in practice. For SED-ML and Kizao, this is different. At the moment, SED-ML doesn't provide you with many parameters needed to tune your simulation algorithm. If you use stochastic algorithm, you need information for the stepping, the time steps, and according to some, it really depends. If you have an algorithm that sample time at equivalent every microsecond, every millisecond, it's not important, but some adaptative simulation algorithm need more information. This information will be provided by Kizao ultimately. So we are moving toward such a situation. That's the ideal situation in my opinion. There is a strong reluctance to that because there is a trade-off between clean systems and working systems, and in systems biology, we succeeded because we had a very pragmatic approach. But as a result, we're still struggling with bad design put in SED-ML 10 years ago. So we have to find the right balance.
So my comment is that listening to this morning's session, the one thing that comes across very strongly is that despite the need for unification, there's this huge spectrum of stuff, right? Everybody sees a need for doing a different thing. And so my question for you is, because you've surely thought about this a lot, so what do you think is the deep reason why it's not perfectly obvious to everybody that there's one good way?
I think, so first of all, there are plenty of things around, but there are not plenty of things that work who have users. A good example is SED-ML versus SED-ML. SED-ML, well, that's actually answered the two parts of your question. In 1998, people developed SED-ML. It was a private company called Physiological, and when we met to develop SED-ML, we actually considered using SED-ML because they have the right way. But it was impossible to implement it. So we choose to use another way. 10 years down the line, it's still not possible to implement support for SED-ML properly. So I guess this is really a struggling point. People kind of agree on the perfect solution. It's not, I mean, we know how to do things, right? It's been done by computer scientists, people doing a large database, description language in other domains, but there is a good way and there is a feasible way. And I think people are more struggling, not so much on the ideal way, but are struggling on the, what will be a likelihood path toward this ideal at some point in the future. But at the moment, there are no many markup language to develop to encode models that have users, right? I know only two of them, as far as I know. I mean, SED-ML and NeuroML.
So in the second part, just to go on a little bit, do you think it's possible that it's in the very nature of science that you can't do this? That there is no perfect answer? I mean, do you think that it's in the nature of the way science is that this desire to have a single unifying approach to it might not be successful in its root?
I think first the word science is probably ill-chosen. I'm just in the middle of a big fight on Obodiscus mailing list with that. There is scientific research, and there is a technology to support scientific research. And the requirement and the feasibility in both domains are pretty different. So that's the first thing. Second thing is, I don't think I'm proposing one approach. On the contrary, I'm not quite on the same chapter than shown here. What I'm saying is that we have all those languages that are perfectly tuned to their purposes. What we have to do is to develop them in collaboration according to the same SOP, standard operating procedure, and the same community guidance so that they interoperate fluently and for the great good.
So just to extend, and hopefully not too long, Rodney's sort of challenge, I think that the impression that you're arguing that there is one perfect way and it should be as follows, or at least there's a framework that's the right framework, is not really the question or the main topic. What you've phrased for us is the challenge of the reproducibility of simulations, or at least models, the portability. If we're not moving towards some frameworks and agreements that allow scientists to represent what they've done, then we're leaving the paradigm that most of our knowledge in science is based on. That's the ability for someone to reproduce the result. if I didn't muddle that too much. So if there's something about the correctness that you're describing, it's that the correctness is to come up with a strategy, to come up with an agreement such that the details of how something was conceptualized and coded can be picked up and redone by others. And what you've described is the best available framework and agreement so far for how to do that. Is that?
It's one agreement so far and one effort that is working. Is it the best or not? Again, I think this is to be put aside. But I entirely agree with you. This is the main point. I mean, a lot of results from modeling and simulation in papers are resulting from artifacts in simulation algorithm. I mean, in the field of chemical kinetics, it's the first page of any textbook show you how you can generate wrong result with the wrong simulation algorithm if your iteration step is not correct and so on and so forth. When you come to computation and neuroscience, the iteration procedure is embedded in the software. So, but when you get different results with two different software, because of a different simulation algorithm, one is better than the other. One is closer to reality. So we have to be able to exchange models so that, and simulation description so that we can objectively say this software is better. So this is something I didn't touch because that was not the topic at all, but Eric mentioned the ecosystem. So now you have several effort groups that are maintaining competition between software. So each time we release a new version of biomodels database, they run all the models on each software, and they basically tells people, okay, this software can support that many % of the model and give correct result. And that's something that brings us a level of objectivity and also a way for the user to choose that is currently still not here yet in computational neuroscience. So, yeah.
Okay, thank you, Nicolas.